Drug-target interaction. Enzymes are biological molecules, proteins, that act as catalysts and help complex reactions occur everywhere in life. Proteins which are crucial to communication system in the body are called receptors. Carrier proteins carry polar molecules across the cell membrane. Nucleic acids have coded genetic information for the cell. Lipids and carbohydrates are structural parts of the cell membrane. Catalytic action of enzymes. Enzymes have an active site. This is part of the molecule that has just the right shape and functional groups to bind to one of the reacting molecules. The reacting molecule that binds to the enzyme is called the substrate. Two major functions of enzymes. 1. Active sites of enzymes hold the substrate molecule in a suitable position so that it can be attacked by the reagent effectively. Substrates enzyme bonding can be due to either ionic bonding or hydrogen bonding or van der Waals interaction or dipole-dipole interaction. 2. The other function of an enzyme is to provide functional groups that will attack the substrate and carry out chemical reaction. Drug-enzyme interaction. Drugs which can prevent the binding of substrate or can inhibit the catalytic activity of the enzyme are called enzyme inhibitors. Drugs which compete with the natural substrate for their attachment on the active sites of enzymes and block the active site of the enzyme are called competitive inhibitors. Some drugs bind to a different site of enzyme, which is called allosteric site. This results in the change of the shape of the active site in such a way that substrate cannot bind to it. If the bond formed between an enzyme and an inhibitor is a strong covalent bond, then the enzyme is blocked permanently. Receptors as drug targets. Receptor proteins are embedded in the cell membrane with its active site projected out of the surface of the membrane. The active site of the receptor opens on the outside region of the cell. The message between two neurons and that between neurons to muscles is communicated through chemical messengers. These chemicals are received at the binding sites of receptor proteins. To accommodate a chemical messenger, shape of the receptor site changes. This results in the transfer of message into the cell. These receptors show selectivity for one chemical messenger over the other because their binding sites have different shape, structure, and amino acid composition. Drugs that bind to the receptor site and inhibit its natural function are called antagonists. Drugs that mimic the natural messenger by switching on the receptor are called agonists.